Okay, so I wanted to take a few minutes to explain a couple of things to you, show you a couple of animations, and explain and make sure you understand how nerves are sent. And so this is just kind of a small section of the notes and the content for how nerves are sent around the body. But this is the most complicated section, so I wanted to make a separate video. One, not knowing quite how long this video will turn out, just because there's some complicated ideas to explain, but also I want to show you how to make some drawings that you might need to know. So here are the three main ideas that I'm going to talk about. One, the first is to define resting potential and action potential, and I know that you can define those things straight from your book. So I actually wanted to not go over what the exact definition is, but rather explain a couple of ideas so you better understand the definition, so that you'd be able to explain them. And then to build on that, I just wanted to add that what we would be doing is using this understanding to explain how a nerve impulse passes along a non-myelinated neuron. And we'll talk about what these things are in case you didn't get that from the notes. And the last thing is to explain the principles of a synaptic transmission. So I listed a couple terms down here underneath resting potential and action potential, but actually some of these terms are related to the second bullet here. So we got to define a couple of things: neurons, dendrites, axons, myelin sheath, and the terminal, as well as the process of active transport. So that's active transport is going to be, we're going to talk about that more explicitly in the next unit, but it is important to understand in this section because it is this is a classic example of active transport, and you need to understand it in order to understand the idea of how a nerve impulse is passed along. So I'm going to go ahead and, I already started this drawing over here. So if you don't have this in your notes somewhere, I'd go ahead and jot this down. I've drawn it, but remember that IB says draw, and when they mean draw, they actually mean draw and label. So I'm going to go ahead and pick a color that I don't have already in my drawing and label a few things. So I'm going to use, let's say, orange to just be very bright. Okay, so if you haven't drawn this, go ahead and jot this down real quick. So again, so I'm going to label this up here. This is why is this not working? Okay, here we go. So this is a neuron, which is also called a nerve cell. And neurons, and you got this from your reading, or you will get this from your reading. A neuron is essentially a nerve cell that's the kind of cell. It's a specialized type of cell in our body, and we have a couple of labels that we need. Actually, I'll do a different color. So that's the main label. So this right here is the cell body. These things that shoot off of it are called dendrites. This long tube that goes all the way down is called an axon. This is just a break in the axon. So think of, like, have you ever seen a graph where they don't label everything on the side? They do a break so they can get to higher values. That's essentially what this is. This axon goes on for a long time. Instead of drawing a really, really long neuron, I've just broken it here. And then what's in yellow here, each of these yellow things, they actually wrap all the way around the axon. And these things are called, all of them together is the myelin sheath. Think of like the sheath to a sword. It's what a sword sits inside of to protect it. And this myelin sheath is made up of, each one of these yellow cells is called a Schwann cell. And you might remember the name Schwann from 9th grade when we talked about cell theory, or when you learned about cell theory. Schwann was a zoologist who looked at animal cells, and these are named after him. But these Schwann cells are made of of lipids. So essentially they're fatty. And I guess this is just more of on a personal note, you know this already, since I have a baby on the way. I often think about this because in the last trimester, when there's a lot of brain development in a fetus, there are strong cravings for really fatty foods from mom. And the reason that a mom wants or craves these fatty foods is so that mom can digest those fats, absorb those fats in the small intestines into the lacteal, so they get spread around the body through the transport system. And then in the fetus, those monomers of fat get put together here around the axon, and we'll talk about what this does in a few minutes. But essentially it helps develop these neurons that are in the brain or throughout, even in the peripheral nervous system of an infant. Okay, so we got this myelin sheath, and then over here on the end, we've got terminals. Sometimes they're referred to as axon terminals, but think of terminal as the end. I always think of these as like airport terminals. If you're looking at a map of an airport, each of these might be a wing in which, gate A37 is down here somewhere. So it's the end of the path. And so I'm going to add one more thing to this label, and the arrow that shows the direction of a nerve impulse. And so just as a quick idea for context, this is one neuron. We've got millions of neurons in our body, and they have to communicate to each other. But right now we're focusing on how a signal gets sent from this end from a dendrite, where it picks up a signal, a chemical signal from this end of a dendrite, or this end of a neuron, and converts that to an electrical signal that gets sent down the axon this way, down to the terminals, and then gets converted back to a chemical signal to be sent from this neuron onto the next one, which then comes back to the next neuron's dendrite. But in this case, what you're asked to do is draw a motor neuron. So what we think of usually at the end here is what we call an effector cell, an effector, which we can think of as muscles. So effectors are things that can cause movement, and those are our muscles. So when you move your muscles, when your heart beats, all of those things are sent from nerve signals that hit effectors, which are your muscles, okay? So let's go double check that we hit all of our terms. We got neuron, dendrite, axon, myelin sheath, terminal, and active transport. So I'm going to scroll down. All right. So we got to talk about active transport. But before I do that, I actually want to talk about diffusion, because we've alluded to that already, and we've talked about its definition, but I want to make sure you have it written down somewhere. So diffusion is simply when particles move from high concentration, that's my abbreviation for concentration, high concentration to low concentration. Essentially, things are spreading out, and this does not have to be across a membrane, because this goes down a concentration gradient. Okay, so this is it's just spreading out. It doesn't need a barrier to prevent mixing. Whereas active transport is when particles move in the opposite direction from low concentration to high concentration. So things aren't spreading out, particles are getting more concentrated. And it does require a membrane. And the reason it needs a membrane is it needs a way to prevent molecules from bumping into each other as they do in diffusion and spreading out. And so we need to be able to contain them so we can concentrate them. So it does require a membrane. And instead of going down a concentration gradient, we say it goes up or against a concentration gradient. Okay, so that's actually quite interesting for this neuron or action potential. So I'm going to zoom in on an axon, not actually zoom in, but in my drawing. So I'm going to scoot over here. I'm going to use that same color green. So let's label this as the axon. So this is what I'm moving the cursor in. That's the inside of the axon. This is the outside, and this is the outside. I should label that. So this is outside, inside, and outside. Now, when we talk about the term of resting potential, we're talking about the potential. I guess we can think of that as potential energy, but really it's the potential voltage charge or electricity that a neuron has when it's not sending a signal, or instead when it's resting. And what you should know is that the inside is relatively negative to the outside. So we label it with negative charges. And we'll talk about this in just a minute. But it's negative to the outside, so we say that the outside is relatively positive. Now, when we talk about relative, what we're actually talking about here are two different ions. We're talking about sodium ions and potassium ions. Now notice that both of these, and if you remember from chemistry, both of these are on the left-hand side of the periodic table, which means that they're more likely to give off electrons, right? So they're going to sit as positive ions because they have more protons than they have electrons. Well, these positively charged ions, well, we might have a hard time understanding why these positively charged ions cause a relative positive or negative charge. So we're going to pause for a quick second. I'm going to show you an animation from McGraw-Hill that explains what's going on to cause this difference. But in essence, what happens is that there is a protein that sits in the cell membrane that kicks out 3 sodium ions and takes in 2 potassium ions for every turn. So if this process, this protein exists all the way everywhere on this membrane, on both sides, top and bottom, and all around the entire membrane of the cell, of the axon, it's pumping out more positive ions than it is taking in positive ions. So we have less positive in the middle or the inside of the axon than on the outside. And when that happens, it's causing a relative positive charge on the outside because there are more positive ions than on the inside. Okay, so let's hang on just a quick second. Okay, so I apologize. So the video that I'm going to show you, the animation, is going to show you what causes a resting potential, but also how an action potential is different, how it's caused, and how it propagates a nerve signal. So let's watch that, and then I'll address a couple of ideas in it, okay? So let me go ahead and pull that up. Okay, so let me see if I can get the mic closer to the computer so you can hear this a little better, okay?
A nerve impulse is an electrical current that travels along dendrites or axons due to ions moving through voltage-gated channels in the neuron's plasma membrane. Voltage-gated channels open and close in response to an electrical voltage. so they are affected by changes in electrical charge around them. When a neuron is at rest, a charge difference is maintained between the inside and outside of the cell. This charge difference is produced and maintained largely by active transport using sodium-potassium pumps.
Sorry. I hit the wrong button. Okay, so I just want to rewind it real quick. So what this guy's talking about is this example of active transport of concentrating things on one side of the membrane over the other, and preventing essentially diffusion of these ions. Okay, so he's talking about the sodium-potassium pump.
Maintained largely by active transport using sodium-potassium pumps. The pump sends sodium ions out of the cell and bring potassium ions in. While other channels allow some flow of potassium ions back out of the cell, the sodium ions cannot easily get back in to replace the lost positive charges. The overall result is that the exterior of the cell has a net positive charge and the interior has a net negative charge. The difference in charge between the interior and exterior of the cell is called the resting membrane potential. A nerve impulse begins when a stimulus disturbs the plasma membrane on a dendrite, causing sodium channels to open. Sodium ions flow into the cell, lessening the charge difference at that location. If the change is enough, it will cause nearby voltage-gated sodium channels to open. This allows so many sodium ions to flood into the cell at that location that the membrane there is depolarized, with the local region inside the cell having a net positive charge and the outside of the cell having a net negative charge.
So what he's starting to explain here is an action potential. So he explained why the inside was negative relatively to the outside, but as you can see here in the diagram, the inside is relatively positive to the outside, and again, that's because these sodium ions flooded in because there was a stimulus, which we'll talk about in a few minutes, that caused a change in these proteins to open up to allow sodium to flow in and depolarize the cell. So it changed the charge inside the cell. It was no longer polarized with positive and negative on either side, but now we're starting to see some equalization of the charges and, in fact, a little bit of overcompensation.
This affects neighboring voltage-gated sodium channels, which then open, moving the depolarization along the membrane. This moving depolarization is called an action potential. Changes occur behind the action potential to restore the resting membrane potential. The voltage-gated sodium channels close and voltage-gated potassium channels open. This allows a rapid flow of potassium ions out of the cell, repolarizing the membrane so that the inside is again negative and the outside positive. This is followed by use of sodium-potassium pumps to fully restore the resting membrane potential and to reestablish proper concentrations of sodium and potassium ions inside and outside the cell.
Okay, so let's go back to this. Okay, so I hope you got that. This is the resting potential. And the action potential is caused when a stimulus, remember the nerve signal for our diagrams are going to the right, so along the axon. And so on this side over here, we would see a stimulus that changes the sodium gates or sodium proteins in the membrane to allow sodium to rush in, thereby changing the ratio or the relative charge on the inside and overcompensating so that it actually becomes positive, relatively positive on the inside than the outside. That change in electrical charge actually changes further sodium channels, sending the signal further and further to the right in this diagram towards the terminals, the axon terminals. But immediately after the action potential, there is a repolarization that occurs. So action potential is the depolarization, or reduction in differences in charge across the membrane, but there is a potassium channel that opens up after the action potential to allow sodium out of the nerve to get the relative charge. But one problem that you might have figured out was that for a resting potential, we've got more sodium on the outside of the neuron, which means less potassium. So that means that there's more potassium on the inside. But immediately after an action potential, and this is after repolarizing to get the same charge as the resting potential, we have the opposite. We have more sodium inside and more potassium outside. And if you notice, it's the opposite of what our actual resting potential is. And so the solution to that is to use the sodium-potassium pump. This is that active transport process that pumps 3 sodium out and 2 potassium in to restore the proper ions on either side for a resting potential. Okay? And that continues over and over and over. Okay? So I hope that makes sense to you. So I'm going to go back up to our objectives here. I think that we've got this first one down now. And I think that we've got, well, okay, let's talk a little bit about this one, the second one, before I put a check mark on it. So I'm going back to this diagram, and what we essentially talked about was how an electric signal is being sent down the axon here to the terminals through having a resting potential, changing that to an action potential by depolarizing and then repolarizing using this potassium channels, which I have a model of, so I'll show you guys in class if you remember to ask. I'll show this to you again and explain how this fits in. And then the sodium-potassium channel readjusts the ions to have them in the proper place for the next action potential, the next signal to be sent. This myelin sheath is very, very important because what it allows the signal to do is jump over the myelin sheath into what we know as this space right here is called a node of Ranvier. It's an R. Ranvier. And it allows it to jump, which means that the signal gets sent a lot faster. So if we didn't have this myelin sheath on these cells, the signal would take much longer to get down from the axon, from the dendrites down to the axon terminals. Okay? So that's what the myelin sheath is for. And so when we were talking about with sending a nerve signal across the membrane, what we were just watching was for a non-myelinated sheath. You need to understand that having this myelin sheath allows the signal to jump over these large sections and travel faster, which is really good for when we think about quick reactions to things or quick thinking, sending signals around your body, responding to stimuli very quickly. Having this myelin sheath is very, very good because milliseconds might make the difference between damaging cells or not. Okay, so let's go back over here and see if we are, I think we can do this now. Now you need to understand the principles of the synaptic transmission. So really what we're talking about is what's happening right here at the end, at the axon terminals, or in other words, what's happening right here before the dendrite of another neuron. So think of this diagram of a neuron lined up, one neuron after another after another. Okay, so let's go down a little bit. I think I've got some space still, a little bit. Okay, so I'm going to draw as best as I can. So let's say that this is so this is an axon terminal, which would make this, well, I guess still part of an axon terminal, but further down here is the axon. I guess I can erase that. So the signal is being sent, again in our diagram, from left to right. And let's say that we've got, let's see if I can draw this better. I'll do it in a slightly different shade so you know it's a different cell. So we're much more zoomed in, but this is a dendrite of another neuron. So this is what we're talking about here, is the connection between one neuron and the next. So we're not talking about it effectors or muscles, we're just between two neurons. And so let's label this space as a synapse, what's also known as a synaptic cleft. But what we find is that the action potential that comes down a neuron from left to right in this diagram actually triggers a chemical that's called neurotransmitters, which you might have heard of before, and we'll talk more about. But these neurotransmitters flow in these vesicles. These vesicles find their way to the cell membrane here at the axon terminal, which go through exocytosis. So this green line that I've drawn, which represents the membrane of a vesicle, will fuse with this cell membrane, spitting out the contents that are within each of these vesicles, which we are calling neurotransmitters, we'll talk more about, and spits them out into the synapse. And they're floating around, and over here on the other end are proteins waiting to catch them, to catch these neurotransmitters. And when they catch the neurotransmitter, what they do is fold in through exocytosis into these vesicles, and these neurotransmitters are considered stimuli to cause an action potential along the neuron, along the axon of the next neuron. So this idea that we were talking about over here, and here, is what is caused by the synaptic transmission along a neuron, or between neurons. Okay, so that's the idea that you need to be able to explain. And there are lots of different kinds of neurotransmitters, and we'll learn a little bit more about that when we get to our option E, which is on neurobiology and human behavior. Okay, and I think that squares us off for these three objectives or statements. And I'm going to go ahead and start another podcast now that's going to be more of a PowerPoint to talk about how these things fit into these concepts fit into our nervous system. Okay, so I think that's done.